So what this figure is showing you in A is at the level of the nerve. So you have a sensory nerve here in your finger. It gets stimulated by something. That pathway travels all the way up your arm into your spinal cord, up to your brain, synapses somewhere in your brain, which gives a message to send a signal back down motor pathways from the brain to the spinal cord, from the spinal cord out to the muscle here, which will then tell your muscle to move your arm away from that stimulus. All right, if we look at it at the level of the actual neurons involved, here we have skin here and nerve fibers that are within the skin. They could be Merkel discs, they could be pain receptors, they could be Pacinian corpuscles or Meissner's corpuscles. Send the signal up this neuron, and because of here, this is a unipolar sensory neuron, afferent, meaning going towards the spinal cord, goes into the spinal cord, makes synapses here, which would take part in a reflex arc, but also goes up the spinal cord, makes synapses with other neurons that make its way to the cerebral cortex, makes other kinds of connections. This is a very simplistic model of what happens. And then those connections will send messages to motor neurons down the spinal cord, make synapses within the spinal cord, and go to muscle fibers. This would be one motor unit, which would contract those muscle fibers and move the muscles according to the stimulus that was received from the sensory neurons. So really just one big circuit that starts with some input, gets processed, and then leads to some sort of output. So remember that when we're talking about nerves, a nerve is just a bundle of a bunch of individual axons within that nerve, and some are going up towards, some are going afferently, and some are going efferently, all within the same nerve. And so when we classify nerves as either being sensory in nature, which means that all of the axons within that nerve are transmitting info to the brain and the spinal cord or the central nervous system. They could be motor nerves, which transmit information from the central nervous system, brain or spinal cord, to muscles or glands to have some sort of effect. Or the nerves can be mixed, meaning that you have both sensory axons and motor axons traveling in both directions within that nerve. To have their respective effects. Examples of mixed nerves include cranial nerves and spinal nerves. Another way to classify nerve fibers is what they're doing. So when we classify nerve fibers as somatic afferent, this means that they are coming from the skin, and afferent meaning going towards the central nervous system. So inward from skin and muscle receptors going towards the central nervous system. If they're somatic efferent, that means they're always going outwards towards skeletal muscles. And because they're efferent, they're going away from the central nervous system. So the central nervous system decided what to do, and now they're going to signal an effect. If it's a visceral afferent, that means it's a sensory signal coming from internal organs or blood vessels, going towards certain areas of the central nervous system, brain, spinal cord, to have an effect. And therefore the visceral efferent would be the motor portion of the visceral. So motor impulse is going out towards smooth muscle, because remember, smooth muscle is involuntary controlled, and our internal organs, we cannot control them by thinking about them, therefore involuntary. Or glands, and glands, again, we can't think about secreting hormones from a gland, and the gland will do it. So they have to come from visceral efferent fibers. This would otherwise be known as your autonomic nervous system. Whereas somatic fibers would be known as your somatic nervous system. In other words, skin and skeletal muscles. I know your crash course video went over the neuroglial classifications and what neuroglial cells are and what they do, but I just wanted to mention one brief thing about microglial cells. Aside from the fact that they act like the macrophages, and this is just an abbreviation for macrophages, they act like the macrophages of your immune system, but under pathologic conditions, such as when the body is undergoing some sort of inflammation, remember you have the blood-brain barrier. So the inflammatory response of the rest of your body does not happen in your brain, but some of those immune chemicals can communicate with the microglial cells in the brain. And when the brain gets inflamed, what happens is these microglial cells will then start attacking synapses. And when it starts attacking synapses, you can imagine there is a whole host of problems that can result. Some of them are result in depression. This may be provide some explanations for schizophrenic conditions and other psychiatric disorders as well. So new research that's coming out on that within the past probably 5 to 10 years. All right, the one thing your crash course videos did not go into as far as neuron structure is the specificities. So what I do want you to get that they didn't cover in there is that the beginning of an axon is this enlarged region right here that's called the axon hillock. And it's in this axon hillock where you have a large concentration of voltage-gated sodium channels, which will start the action potential when they get triggered. If we follow the axon all the way down to the end, we come to the synaptic terminal, otherwise known as the synaptic button or synaptic knob. And in between the synaptic terminal and the receiver of the signal from the synaptic terminal is something called the synaptic cleft. There is always a small gap between the synaptic terminal and the next cell that's being stimulated, across which neurotransmitters can be released. All right, so when these neurotransmitters are released, what they do is they bind to receptors on the postsynaptic membrane of the target cell. And usually when these neurotransmitters bind to these receptors, these are ligand or chemically gated channels. They'll open up and allow an ion, usually sodium if it's an excitatory synapse, to go through to depolarize the next cell. All right, so the difference between gray matter and white matter in your nervous system. Gray matter tends to consist of cell bodies and dendrites. So gray matter is receiving information. And white matter tends to be myelinated axons. And because myelin is a fatty substance that covers the axon, it's going to have a fatty appearance when you have them en masse in one location. So white matter is going to be sending information. All right, gray matter receives information, white matter sends information. And in the brain and spinal cord, you'll find that the spinal cord has its gray matter, in other words, where all the synaptic connections are made, all the information is received, is on the inside of the spinal cord, but all the white matter is on the outside of the spinal cord. All right, so information comes in, makes a synapse, and then goes into the spinal cord, the white matter of the spinal cord, to go up to the brain. All right, if you go up to the brain, you have to have a pathway to get into the brain. So all of the white matter is going to converge into the medulla through the pons and through the midbrain until then it branches out to make connections in the cerebral cortex. So all of that white matter that was on the outside of the spinal cord is now feeding into the brain through the central portion and then spreading out so that the gray matter in the brain is on the outside, but the white matter is on the inside. And so I hope that makes sense to you. Think of it as wires coming up from the spinal cord, going in through the middle of the brain, and then going out and making its connections on the periphery.